Hello, everyone. Thank you, Jamie. Hello, everyone. It's my pleasure to introduce for a second time Dr. Makoto Miyakoshi, who is going to be talking to us today. The title of his talk is on EEG generative mechanism. And Makoto has said that if there are things that are not clear or burning questions that you have, that you should please interrupt him. That's right, Makoto, right?
Yes, yeah, please.
Okay. So Makoto, the floor is yours.
Thank you. So let me share my screen. Okay, and start my presentation. Okay, do you see my slide?
Yes, perfectly.
Okay, thanks. Okay, so today I'm going to talk about the generative mechanism of EEG. And I think having this insight is very important because if we have that good understanding, that means our eyes are well theory laden. And if we have a theory laden eyes, we can see the phenomena through the generative mechanism. So that is the same as we see the phenomena from the standing point of the forward model, like the beginning of the forward model. And we are seeing the phenomenon as a result of the assumed mechanism. So let me explain what it means. So this is the classical ERP of the mismatch negativity by Natanin almost 45 years ago. And we see the difference in the ERPs. And this difference comes from the fact that the standard is the ERP response to the and the signal is a this is a deviation from the background context. So that generates the ERP difference. Great. But what do we see here? What kind of brain mechanism can you predict from here? Or what is going on inside the brain? What is the brain correlate of, or whatever this electrophysiological correlate of this phenomenology in the ERP difference? And this is a part of it, right? The EEG is generated as a result of the interaction between the thalamus and the cortex. And thalamus has reticular nucleus, and the reticular nucleus works as an inhibitory mechanism. And the cortical layer has 5 or 6 major layers, and those layers have receives the differential the modulation and control by the different thalamic nuclei, and those layers generates the different the activities, which is studied by the Lakatosh recently. So there is a almost 40 years difference between this Natanin study and the Lakatosh study in 2020. So this is an inverse problem we see. So if we can explain the background activities like this, including all of these mechanisms, and then we can say that we have a good understanding of well, it's not perfect, but it's still a good explanation we have. And if we have this, another thing we can do is to make the forward model. So that is how we build the similar machine or mechanism that generates the ERP-like phenomena. So this is one example of such a forward model called HNN, Human Neocortical Neurosolver. So we basically simulate the cortical mechanism and thalamic inputs, and eventually this simulation environment generates the oscillatory, EEG or MEG signal-like output. So using this forward model, we can make more interesting studies. So that's the benefit of having a good generative model in our mind. Now, today I'm going to focus on this central question, which is where do EEG rhythms originate from? Because this is also has been the central question of the history of the EEG, at least in the 20th century. So I will go back way more to the point of the end of the World War II. There was one study that showed that isolated thalamus can show the oscillation. And this is a decorticated cat thalamus. Decorticated probably means, the cortex is functionally at least, dissociated. So this means that, okay, thalamus has this rhythm generator. So at least thalamus is one good candidate to show that rhythm, which is called spindles. And the natural extension of that idea is now this Anderson in 1967. They said that, well, actually, I cited one of the sentences here, which contains two important mistakes, later refuted. The first one is that they said any thalamic nucleus was capable of exhibiting rhythmic oscillations. This was not true, actually. Some thalamic nucleus do not show rhythmic activities. Another important the statement they made was that the rhythm generated inside the thalamus would then be imposed on the cortex via thalamocortical cells. So this idea is probably still believed by some of the EEG researchers who do not have, who haven't read those studies on this issue. But this is as if the thalamus is the only rhythm center, and the cortex is just driven by the thalamus. But recent studies showed there is interactions between the thalamus and the cortex, and also cortical-cortical connection can show some rhythmic oscillations. By the way, on the right side of the panel, I show the tractography of the thalamocortical radiations. This is basically covering the entire cortex, not surprisingly. So this means where cortex is seen, there is thalamic connectivities, and different thalamic nucleus have different cortical controls, as shown in this Piper 2022 paper. Now, I want to start with this a series of studies by Sterriade, and he focused on the spindles. And spindles is it's a form of oscillation, and he found that one of the important thalamic nucleus, the reticular nucleus, has three oscillatory modes: delta, alpha, and gamma. And this rings a bell to us, right? It's like, aha, we know these frequency bands. And he also found that this 7 to 14 hertz, which is practically the alpha band, is associated with the resting membrane potentials. So this means that if we observe the 7 to 14 hertz oscillation in this reticular nucleus, then via the thalamocortical the connection, the EEG power at the cortex should be reduced, right? So there must be such anti-correlation between this reticular formation and the cortical electric activities. So that's the prediction. And later, using the PET, the Larson and colleagues showed that indeed, when they observed the increase of the regional cerebral metabolic rate within the thalamus, then the simultaneously recorded EEG power dropped. So the blood flow increases in the thalamic region, then the EEG power went down. Particularly the alpha power went down. So this reticular formation is an inhibitory mechanism of the thalamic activity. So this is the increase of the reticular formation activity is putting the foot on the brake, right? And that I'm not good at this double negative, but anyway, yeah, this disinhibition and this process is here. But later, there was another important findings made by Lopez da Silva, and he said the well-studied spindle phenomena is actually not the same as alpha, and the reason for that is, number one, Sterriade and colleagues used cats, but Lopez da Silva said actually cats are not a good animal model for the human EEG, and actually dogs are much better as a human EEG model because the alpha rhythm they showed is similar to the human's alpha rhythm. And also, this spindle phenomena were mainly studied during the sleep or anesthesia. But the alpha rhythm during sleep is not very dominant, right? So the sleep or anesthesia is associated with the very low consciousness level, but usually alpha is observed during the awake state. So those two different experimental situations, and in fact, when the authors compared the actual recordings of the alpha and spindles from thalamus and recorded from the thalamus and cortex, they showed different peak frequency powers. So they are not the same. And later, using the MEG, these authors showed that the source of the spindles and alphas and central mu are all different. So alpha and spindles are unfortunately two different things. We have to admit that. The same author, the Lopez da Silva, also found in different papers that actually the source of the alpha oscillation is in a cortical laminar structure. What he did here is he used these depth recordings, and this needle electrode has maybe 3 or 4 millimeters in length that penetrates the 6 layers of the cortex. And the superficial layers, those electrodes shows the peak along with this axis, but this peak gradually shifts its phase to become the trough. And when he plotted this phase difference in this way, then he found a sudden change from between electrode 23 and 22, almost 180 degrees polarity difference. So this means that there is a positivity here and there is a negativity here, or the opposite. But there is the polarity reversal. This indicates that there is a dipole, current dipole here. So this alpha is actually generated inside the cortex, cortical layer systems. This finding was later validated in this way. This is only one of the studies that have validation effect. There may be other good studies which I missed, but this is another laminar structure recordings, and this is from the superficial, and this is from the deep layers. And what we see here is that basically this frequency range here is in a higher gamma band, like above 50 hertz to up to 200 hertz. But this gamma band activity is temporarily organized in a low frequency way, like up, down, up, down, up. And this up, down rhythm is in a delta frequency. So this is a typical example of the phase amplitude coupling. So this phase is defined by this low frequency delta, and that regulates the activity of this high gamma band burst, like brr, 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 brr. And what they found was that this phase amplitude coupling comes from the superficial layers and not the deep layers. So there are those layer-specific activities and modulations, and those layers are later, which I will later explain, differentially controlled by the thalamic connections. This is another example of, and this is kind of rare visualization of the depth by depth visualization. And this shows that alpha source comes from 800 micrometer above the granular layer level. So that means 800 micrometer higher than this middle granular layers. And gamma source is from the minus 300 micrometer. That means deep. So this alpha source and the gamma source are differentially modulated and generated in different depths of the layers. And again, the different depths means different thalamic controls. So let me summarize the difference between the spindles and alpha. So spindles were studied by steroid and recorded during the anesthesia or sleep, and they were mostly studied by cats. And they studied very well. So the generative mechanism of the spindle is very well studied. Actually, that's a great advance. On the other hand, the alpha, which is what we observed in the EEG studies, the Lopez da Silva pushed these studies and recorded during the awake period, and mainly dogs were used. But the cellular mechanism of the alpha generation is actually not very well known, at least at the point of 1990 when this paper was written. So here is one unfortunate thing for us EEG researchers. Spindles are not alpha, but spindles are much better studied. But we want to know alpha. But how can we apply our understanding of the spindles to the alpha? That's the question. Because again, the alpha was not as well studied as the spindles. By the way, I found one interesting thing when I used PubMed to search the word alpha spindle, which is a very confusing term. If you think about those differences between the spindle and alpha, and how long these researchers spent on differentiating those two concepts, and the term alpha spindle seems to appear after 2009. That's like 20, 30 years after those authors studied these phenomena very well. So I think this means that the EEG researchers forgot about this difference, and we are getting back to the pre-steroid Lopez da Silva era, and we don't know the difference between spindle and alpha. So this is how the valuable research results were become forgotten by the next generation. So we have to remember those great studies. So the conclusion at that point is that the spindle can be the model of alpha. So even though the spindle is not exactly the alpha itself, but because spindle is very well studied, it should serve as some kind of model of the alpha. So that's an analogical thing, but not exactly the alpha itself. So this concluding remark was made in a 1990 paper, which is supposed to be one of the most cited references in the field. So thalamus drives and mediates cortical-cortical coherence. And this is again the study by Lopez da Silva in 1980. So this is I think, one of the most important studies in the history of EEG in terms of this thalamocortical connections. What he did is this. So this is a dog, the cortex, and there are two recording sites, electrode 8 and electrode 15. And when he measured the ordinary coherence between these two electrode locations, and this is the power spectrum density of the coherence. Not the power spectrum density, but coherency. And not surprisingly, there is a peak around the alpha frequency band. This is ordinary coherence. Now, he applied the partial coherence, and the partial coherence means the coherence between, this is still the coherence between the electrode 8 and 15, but the difference is this is after subtracting the common influence from electrode 13. So this is like a residual after subtracting the common influence from the electrode 13. So if this is the residual, okay, if we don't see much alpha peak here, it means that this ordinary coherence peak at the alpha range is actually the common input from the electrode 13. That's why after subtracting this out, you don't see much. So that means maybe the electrode 13 is sends a common drive to those two electrodes. That's why those two electrodes were synchronously activated. So using this computational tool, what they did is that now they evaluated the cortical-cortical connections, but taking into account the thalamic input, which is the lateral geniculate nucleus in this case. And surprisingly, there is not much difference between these two. This means that lateral geniculate nucleus has little to do with this cortical-cortical coherence. But what they found was that it was actually the pulvinar that co-modulate the cortical-cortical connectivities at the alpha range. So this big alpha peak disappeared. That means the pulvinar sent the common drive these two cortical-cortical recording sites. So this is the same as the theoretical thalamic deafferentation. So that's like computationally cutting the input from the thalamus. Based on these beautiful studies, they concluded that the pulvinar affects the cortical connectivities more than the LGN. And later, there was this study published from Science. And this pulvinar modulation of the cortical-cortical connection was actually the substrate of the top-down attention. So they used the top-down attention visual task using monkeys. And there is a cue, spatial cue shown. And they asked the animal if this cued position showed bow tie or not. And this is ABCD is time frequency decomposition of this data. And you see, between the attention on and off conditions, when the top-down attention is on, then there is always strong pulvinar to V4 Granger causality at around the alpha frequency range. So this is a great validation of the studies. But again, I noticed that those authors, these are apparently the imaging researchers using MRI. They did not cite the Lopez da Silva study in the 1980s. So this is again the evidence of this next generation forgets. the former generation's good achievements. So we have to remember these good studies. So that was a very nice clarification on this aspect of the thalamocortical relations, which is a coupling. So thalamus can control the coupling between the different cortical regions and their activities. Now, because we are interested in clinical studies, then this thalamocortical dysrhythmia is the critical concept for all of us. And one common findings among the variable clinical status, such as depression, Parkinson's disease, tinnitus, and neurogenic pain, they commonly showed the number one, the alpha peak shift from higher to lower frequencies, and also the overall power increase, as shown on this right side panel. And these common features are now referred to as thalamocortical dysrhythmia. And our groups also have reported the very similar findings. Like schizophrenia patients showed lower frequency and higher power alpha and increased gamma. And this is a comparison between the control and Fragile X syndrome children. And they showed a markedly lower alpha frequency and increased power in the peak frequency range and the gamma tail part as well. What I like about this Linus 1999 paper is that this author uses a very impactful word. So as a foreigner, non-English, native English speakers, it's easier to understand, such as, oh, this is all the sightings from the paper, but they said, oh, the mindless burst mode. So when the thalamus mindlessly bursts, then this disturbs the normal cortical activities. Or they also said this pathological state, the thalamus is stuck in spindle-like activities. They also said there is an abnormal low-frequency oscillatory attractor. So once that attractor captures the thalamic states, then it's hard to get out of there. So getting out of there means the disorder is cured, right? But this attractor is, in a sense, in the EEG representation, the associated phenomena of the disease state. And these abnormal thalamic activities can be further connected to the membrane hyperpolarization or type 2 CA2+ channel D inactivation. So there is further underlying physiology of this phenomenon, and that can be pursued by the other researchers. And importantly, some of the deep brain stimulation application or thalamic lesions aim at reducing this some specific part of the thalamus. So this is like a proof of concept by using the disease template to prove that there is specific thalamocortical circuit that can go wrong and that can be cured. So this is the model they proposed in their paper, which is not, in my opinion, super helpful or particularly beautiful, but we have to stick to this figure. And I'm not going to explain the detail of this figure here, but interestingly, this figure has been used repeatedly in later publications by other researchers even. So this is a later validation. This is a sliced study. So they sliced the ferret thalamus, and so conveniently, this slice contains both reticular nucleus and thalamocortical neurons. And they delivered the electric stimulation to the thalamocortical neurons, and they recorded the electric responses within the reticular nucleus. Reticular nucleus is, again, it's a brake on the system. And this is before the electric delivery, and this is the stimulus delivery. So this is the model of deep brain stimulation, basically, using the ferret. And before delivering the stimulus, this is the blow up of the first box A. And under this long spike, you see the, it's actually sinusoidal, but the rhythmic activities here, right? This is the spindles. And this is the generator of the EEG, by the way. So if you further blow up this one of the 3 trains of the spindles in E, then there is a low frequency bump, and a spike is on top of this low frequency bump. So we can see the generative principle of the EEG itself here. So this low frequency thing can overlap very well after averaging. So this can make accumulate and reach to the point that the summary of this EPSP can make a significant contribution to the scalp recorded EEG. And also you notice that the spikes here only occurs at the top of this wave. So this is the phase amplitude coupling. So there is a low frequency phase like boom, boom, boom. And this high frequency brr, brr thing only happens when this low frequency goes up. So this is also the generative principle of the phase amplitude coupling. After stimulus presentation, this pathological spindle is gone. It's silent, right? And after some time, it will be back with the spindles. So from the macroscopic electrophysiological viewpoint, this spindle is a bad thing. But of course, this spindle means this action potentials as well. So these things are clarified and validated later in this way. That Linus 1999, I think, is the explanation about the activity. So when we see the scalp recorded EEG, then that is what's the generative mechanism of that? Well, if it shows the pathological signs, then probably the thalamic nucleus and particularly the reticular nucleus is not working well, so that thalamus is bursting in a mindless way, as Linus said in the paper. There is another interesting property of the thalamus, which helps us to connect the micro and macroscopic spatial scales. In this paper, these authors argue the difference between the matrix cell and the core cell. And core and matrix difference is that this blue is the matrix, and matrix means diffusive. And this diffusive thing goes everywhere in the brain, but the core means it modulates a specific functions such as motor and those some of these specific functions. And interesting fact is that this matrix projects to the superficial layers, and the core projects to the deep layers. And this is the within thalamus distribution of the matrix and core cells. So the anterior part is mainly the matrix cells, and the posterior part is mainly the core cells. And this is the matrix cell projection destinations, and these are the core destinations, core cells projection destinations. And I immediately noticed that this core cells are associated with the brain region that shows a beautiful ERPs, like visual evoked potentials, or whatever this motor related mu rhythm or stuff like that. And matrix cell, well, I would say maybe one exception of my assumption, I mean, the hypothesis is the error-related negativity. You know, the error-related negativity shows a very nice ERP patterns, but overall, you know, this orbitofrontal or frontal regions, I don't expect beautiful phase reset, nice ERPs from these regions. So I thought maybe core matrix difference is also related to the, you know, the ERP observability. And in fact, this core matrix cells, core cells are, you know, have projection to the deeper layers, and deeper layer means the dipole is going to be large, right? So maybe that's why core projection shows a nice ERPs, but I could be wrong here. That's just assumption. So that is also about the coupling, like how thalamus can control the coupling between the neurons or the cluster of the neurons between the cortical columns. So this is another example which I liked, is that this is fMRI studies, I believe, and when they correlated between specific thalamic regions activations with the PCA dimensions of the brain states. So PCA dimension simply means if the brain oscillation or bold signal activation mode has simple, like, you know, a simple mode or complex mode like this. And they found that this the thalamic nucleus shows a strong negative correlation between the PCA dimensionality. So this means that when this thalamus activates more, then brain starts to activate in a more simpler mode. And we can here use the matrix and core separation again. And Gallet, this author, reported this region. So this region, I believe, falls within this matrix cell region. And matrix cell covered the frontal regions, and that I think broadly agrees with this prediction made by the authors, and which probably overlaps somewhat with this dorsal attention network. So we can make this kind of conjectures based on these results. I think that relates to the topology, so how thalamus controls the cortical activities. Although most of these are more or less overlapped, as the authors admit that at least 4 partially overlapping canonical functions. So these separations are not orthogonal. And this is an interesting study from back in the 70s. What they did was to inject some poison took from the pufferfish, tetrodotoxin. And what it does is to silence the EEG, as can be seen here. So this is a baseline EEG recorded from some animal. And this is 10 minutes after tetrodotoxin injection. So you can see the appearance of silent period, and the overall amplitude are somewhat reduced, and that continues for another 10 minutes. And they studied the correlation between the intracellular spiking activities and the EEG. So in this case, EEG is macroscopic recordings, and intracellular means microscopic, right? And what they found, oh, by the way, this is a standard sleep and awake without tetrodotoxin. This is after injecting the tetrodotoxin. What they found was that intracellular activities were not silenced very much. but EEG activities were very well silenced. So if I summarize the point, it's going to be like this. This is an exaggerated schematic illustration. But the point is that the claim is that intracellular activities were less affected, but scalp EEG was very much affected. So they concluded that actually tetrodotoxin affects this cortical synchrony and not the spiking activity per se. And I think this argument is a bit strange, but this is a very highly valued study in the field. I ran also simple simulation using MATLAB, and assuming some of the given values, like each neuron contribute 0.01 microvolt, which was used by EROL. This is a comparison between group of neurons, and they are all randomly activated. And as you increase the number of the neurons, then the measured EEG amplitude increases in this way. And compared with that, this is only 1% of the neural population get activated, but in complete synchrony, like 100% synchrony. And 99% of the other neurons are dead silent, okay? And when you make this comparison, there is a point where this synchrony model goes beyond the total amplitude level of the random neuron firing in this way. And of course, this 1% synchronized is underestimation, because 99% of the other neurons must be still randomly firing, right? So this red line must be more than this, but this is for the sake of the comparison. And according to the Nunez and Srinivasan, we have to have this much cortical area to produce the scalp measurable EEG that is assumed to be something like 5 to the 10 to the 7th neurons. That is well within this region, which is 1% synchrony overwhelms the random mass effect. According to these MEG researchers, they even showed this figure in this paper, and they said that if they actually have 1 square centimeters of the cortex with only 1% of the synchronization, which is this line, then they can measure the MEG signal from there. So this is surprising, right? We talk about, neural synchrony, but with this, the mass effect, only 1% of the synchrony makes the difference. So we don't need to have 50% synchrony or something like that. Just 1% is fine. And because in the case of the EEG, it's not 1 square centimeter, but 6 square centimeters. This means this graph goes far right, and we don't even need 1%. We probably need 0.1% or much less ratio of the synchrony to record the 20-30 microvolt of the EEG on the scalp. So we have to have this basic numbers to have a right insight of the neural synchrony. We shouldn't talk about 80% synchrony, but 0.1% synchrony. That makes a difference. So that's the neural coupling. Yeah, probably I can stop here because this is a good point to stop, or I can move further if you want.
I mean, you have a few more minutes if you want, like maybe 5.
Okay.
5.
So let's
please continue.
go really quick. Okay, thanks. So this is another very important study from the 1965. And this is extremely surprising for me when I saw this for the first time. So they did a very simple study using this piece of the skull removed from the patient. And they put this is the skull, fresh skull wet, and this is a polyethylene layer, which is non-conductive. And they put the punch hole, probably using the needle. And this is the cotton soaked in saline water, and this is the low frequency oscillator. And they measured the electric potential difference here, reference to somewhere else. So what they studied was the difference in the ratio between this generator's voltage and the measured voltage. And when there was only single pinhole, then the measured voltage here was 20,000 times smaller than this generators. And they increased the number of these pinholes to the area, to the area of the 1 square centimeter. Then attenuation rate becomes much smaller, 400 to 1. And when they reached about 1 square inches, then it's 2 to 1. And this is a 10,000 times different. And it was almost incredible. When I presented this in front of my colleagues, no one believed it. And it's very difficult for me at the time to understand why this happens. So by the way, this is the reason why today people talk about this 1 square inches. And this was further later supported by Paul Nunez's theoretical and empirical studies. And these are the surprising examples they showed in the paper. This is a subdural electrode, and this is a scalp electrode. And when the subdural shows this activity, the scalp are pretty much silent. And of course, the question is, why are they not showing a good correlation? And this is a very extreme example. They applied 50 millivolts between these two electrodes. This is a human head, by the way. And this is the EEG recordings. And when they added 50 millivolt here, they measured 10 microvolt difference here. Why is this effect so small, right? Another example is, between these two electrodes, they applied like 850 millivolt. I don't know what happens to the brain at this point, but they only measured like 25 microvolts difference in the electrode right above these stimulating electrode. So this ratio is like 5,000 to 1 or 34,000 to 1. And between the electrode 83 and 84, which is only a few millimeters separate, but there is no volume conduction effect. And why is that? And also the O2, which is the recording from the surface, scalp surface, and 83, 84, there is very poor correlation between the electrodes. So why is that? Where is the volume conduction effect, right? And this is also the very often quoted comment by Gray Walter. He said, when he measured alpha activities, practically adjacent and almost congruent one field, he could still observe a variety of alpha rhythms. So in cortical recordings, this happens. So we have to take it as a fact. And the explanation is this. So if you think about the single dipole, and let's say there is an 8 millimeter, the distance inside the brain. So that is actually this replicating this condition. The electrode positions are 8 millimeters apart, and the recordings are this different. This is only 8 millimeters apart, but electrode 71 shows huge spikes, but electrode 70 doesn't show that at all. So if you apply the standard theory of the volume conduction and the dipole model, it's going to be like this. This attenuation rate is huge, like 200 times or something like that, or 60 to 200 times. And this is the comparison between the on-axis potential and off-axis potentials. And this is, by the way, the simulation I run. So not surprisingly, this on-axis measurement shows much larger potential at around this point, and that gradually decays in this way. And this off-axis is not that big, so there is not much benefit of this off-axis measurement. But because there are a lot of these off-axis contributors in the field, much more than the on-axis contributors, and that ratio is about 1 to 130 in this simulation. So 5-degree circle compared with the 45-degree circle. And there are much more, like 50-degree circle, 60-degree circle is possible. So this ratio is still conservative. So if we think about the dipoles, we should actually think about a lot of dipole, like a dipole layer like this. And even though the on-axis, off-axis difference is about 3 times, because there are 130 times more off-axis contributors, these off-axis contributors can overwhelm the on-axis contributor. So in the end, when you compare the single dipole and dipole layer is so robust against the distance-dependent attenuation when, for example, the measurement point is about 10 millimeters separate from the source. Then if you have this much dipole layer, which is, again, this is a number suggested by Nunez and Srinivasan, the dipole sheet has 26 times less distance-dependent attenuations. So this is a very important generative model of the EEG. So this means that how much voltage we can measure as an EEG actually is largely dominated by how large the source is, not like how strong the source is, how large. And again, the synchronization ratio we observed was, it tells us that it only needs like less than 1% of the synchrony. Okay, those are all the presentations I prepared for today. Thank you very much.
Thank you. That's wonderful. Do you want to stop sharing and we can see if anybody has questions?
Sure.
Great. Well, I'll start.
Okay.
Because I have questions, but I just want to make a comment first, which is that I really appreciate your presenting and citing the classic older work. I think too often we're in situations where it isn't cited, and people publish and as if they're rediscovering or discovering something which really, in terms of these classic studies that you presented, has already been elegantly done.
Yeah, I agree with you. Yeah.
I just have a very specific question. I was really interested in your description of spindles. And EEG. And I was reminded of EEG in preterm and very newborn infants, and the characterization of what are called sleep spindles. And I'm wondering if that characterization in the EEG is what you were referring to. I mean, I know obviously it's scalp recorded EEG, but I was just wondering if it was a similar characterization.
I am not in a position to give that a definitive answer because I'm not necessarily the specialist of this spindle alpha difference.
I do sleep research, and the sleep spindles are what he's discussing, and the thalamic contribution to those spindles, the reticular nucleus has been very well
That's what I thought, Linda. Yeah. But you don't see it. You see it during sleep and in premature and newborn infants, right?
Oops, sorry, it's actually a characteristic rhythm of sleep.
Yeah.
It characterizes non-REM sleep.
Even in newborns?
Well, the newborn rhythms are a little bit different.
I
So
see.
I don't know the degree. So I haven't studied sleep in babies that much. I believe that the spindling still occurs, but the duration and magnitudes differ in the little ones. And I'm not familiar with the preterm literature at all. So mostly I'm talking about adults.
Hey, we have time for a few questions. Anybody?
Hey, Makoto, this
Oh, okay.
Go ahead.
We have Jim and then John. So Jim.
Oh, okay. So here's the question that pops up in my mind when I look into these studies, these 40, 50, 60-year studies of the generative nature of EEG. They tend to all be very descriptive. They tell us that a lot of regions have a lot of roles. I mean, that's great. It's work from 40 years ago. What I'm really interested in is a prediction or a model or a way to help interpret my data. So how can I use these data in a way that can help explain something about cortical ephys that I wouldn't have in another way? And I'll give, one example just from the work you presented that popped up in my mind. For example, work from Max Schein's lab of those different topologies that are related to different thalamic networks. That looks like something you could take a cortical mask of and use as, say, a prior or something for a source estimation procedure, which I've never seen anyone do. And I don't think our field is really trying to take the history and move forward to empirically constrain or predict novel predictions. So if you have any, I'd love to hear them.
So I briefly mentioned about the human neocortical neurosolver in the beginning, but I think making such a forward model and make predictions and make this the generative mechanism as a like a research principle to reproduce the phenomena is one way to go. Although brain is notoriously complex, and some of the smart EEG researchers like Paul Nunes is completely pessimistic about such a forward model predictions. But because, well, it depends on what kind of predictions those researchers have. But if such a prediction is mainly focusing on the non-linear interactions and emerging phenomena, then making a linear model does not help, right? So that kind of pessimism is there. But I still think that starting from the simple model is not necessarily a bad thing in science, right? We all start from the dumb simple model and refines and refines. So HNN is, I think, a good prediction, good direction. And in fact, our lab has been studying collaboration with the Stephanie Jones, the developer of the HNN model. And I'm sure there are many other good models and predictive studies using those the neural models in computers, like in silico neuroscience.
We have time for just one question. John, I see you have your hand up, so.
Thank you, Makoto. That was a wonderful talk. I wish you would give that to all of our residents and neurophysiologists here at Washington University. We have these theoretical discussions all the time that aren't well bounded by data, which is so fantastic that you took the time to think about this. One of the things I've often worried about, I'm a pediatric epileptologist, is childhood absence epilepsy, where you have this massive high amplitude 3 Hz spike in wave activity, or in infantile spasms where you have hypsarrhythmia. We always argue about how many neurons that really is, particularly when it's showing up all over the cortex. We're not just talking about a patch of cortex. Have you thought about this or have any any general thinking on how we should think about these very high amplitude, large areas of cortical activation?
Well, I think it's apparent that thalamic dysregulation is involved here, but also there is such a thing as the global field theory of the brain. And I think those two things must be considered in that case, particularly if you talk about this rather large areas involved, then the oscillation mode of the entire cortex, I think is we cannot neglect. So, but not many EEG researchers are aware of this global field theory. And I believe this is one of the things actually that was, the example of the forgotten, good neuroscience from the last century. So definitely thalamus, but potentially the global field theory. So those are the two things I would consider. And there is also like a nonlinear physics-based studies, like what was the term, like limit cycle model and stuff like that, which I'm not very familiar with. But it seems this a large area oscillation with a relatively low spatial frequency mode that's something single unit recording researchers cannot explain very well, right? Because this is oscillatory mode issue rather than single unit or the thalamocortical specific loop, but it's entire system oscillations. There are those studied in excellent research, but these research are very difficult to understand, because it requires a very deep knowledge of the physics. Oh, that's what I can say. Sorry about it.
Okay, unfortunately, we do have to stop. Listen, this was great.
Thank you.
Really, very, very much appreciate the talk that you gave today. And as I put into the chat, the recording will be available on Confluence for HBCD people. If you're interested and you're not HBCD and you're interested in the recording, just write us and we'll be happy to share the link. Okay. So Makoto, thank you so much again.
You're welcome.
And everybody have a good day.
Bye.